Hello everyone, my name is Víctor López and I would like to explain you this poster entitled Detection of Mesial Networks with Magnetoencephalography during Cognition. So first of all, please let me give you a brief summary of this work. So the question that we want to answer is, can we detect the hippocampal activity with MEG during cognition? So to address this question, we have based our work on 5 points. First is a task of recognition memory. Second, we have used blind source separation analysis on MEG, like independent component analysis, so we can disentangle and separate different sources that mix on surface recordings. Third, we have used simultaneous intracranial and MEG recordings, so we have the same information inside the brain and also outside the brain. Fourth, we have done an analysis of partial correlation between ECG and these independent components. And finally, we have applied source localization on the components that we have carried on MEG topography, so we can confirm the localization of our components on MEG. So the task that we have done is a quite simple memory task of images. So it's divided into two different steps. The first one is encoding part, so we saw to each patient different images, like quite simple images, maybe a dog, a house, a car, things like that. Then we have a short video of 1 minute, it's a distraction video. And finally, we have the task itself, when we present the patient new images. Some of them are new, so we are presenting them for the first time, and some of them are the same that we presented during the encoding part. So we have here two different conditions, the new images in red or the recognition, which are the old images in green. So on our MEG recordings, we have applied SOBI. SOBI is a type of independent component analysis, and we were able to retrieve one component with the same topography in all of our patients. This topography seems to be a deep topography because we have like one dipole where the source and the sink are far away from each other. So it's our putative deep source. And the event-related potential present differences between old and new pictures around 500 milliseconds in 3 of the 4 patients, which is also the time where we expect differences based on the previous work and for the deep structures like the medial temporal lobe, hippocampus, and so on. So with this source, so we have one component for each patient, we have applied the correlation, direct zero lag correlation between the ECG recorded at the same time and between these components. So these are all the electrodes that a scheme of all the electrodes that we have used. And here we have summarized the results. So for each electrode, we have selected the channel with the highest correlation with the SOBI component. Also, we have marked with some stars those electrodes that have at least one channel with differences between old and new images around 500 milliseconds. So we can see that the maximum values are here in TB, for example, also in B, in A, B, TB, which corresponds to the electrode A is targeting the amygdala, B, the hippocampus, TB, the renal cortex, and TP, here, the temporal pole. So the highest correlation is between those electrodes that are targeting structures in the medial temporal lobe. So we have an indication that probably our putative deep source is representing some activity deep from the medial temporal lobe. So here we have a summary of all the different correlations we have between one patient and the different sites that we have on each electrode and the SOBI MEG component. So we can see that the maximum correlation, for example, in this patient is in TB, that, as I said before, correspond to the renal cortex. But we have also some quite important correlation between other areas like amygdala or the hippocampus. So as we have important correlation between all these structures and our component of MEG, so we've applied partial correlation analysis. So we have selected for 3 of the 4 patients, because we don't have all the structure in one of the patients. We have selected 4 structures, the hippocampus, amygdala, temporal pole, and the renal cortex, and we have computed the partial correlation between these 4 structures, and also we have included of course, the independent components of the MEG. We can see that even if the correlation is quite high, for example, in this patient, in the hippocampus, amygdala, and the renal cortex, not so high in the temporal pole. So we have a decrease in the partial correlation, a general decrease in the partial correlation, as is expected. And this is more pronounced in the case of the amygdala compared to the hippocampus, temporal pole, and the renal cortex. And in these three patients, we can see that in 2 of them, the amygdala is not contributing at all to the partial correlation, while the other 3 elements are in all cases contributing. Maybe the renal cortex is the structure that more contributes to the independent component, but also the hippocampus and the temporal pole has an important contribution, and it's a direct contribution. That means that it's not this information is not shared by any other structure. So we have some information that is exclusive in the hippocampus and that we can retrieve it on our independent component with MEG. So here this is an example of the ERP of the independent component, and also the ERP of the left hippocampus of one patient and the right renal cortex. So we can see that in black we have the independent component, and the hippocampus is following the same activity at the beginning, but in the middle it stops following the hippocampus and starts following the renal cortex. So actually that represents that our independent component is a mix of these structures. Actually, it's a network combining the information of these not 4 but 3 structures. As the amygdala probably is not contributing independently to this component. Just the correlation with amygdala can be explained by the correlation between the independent component at any of the other 3 structures. So finally, to further confirm the origin of our MEG component, we have applied a 2 symmetric dipole localization. We have applied 2 symmetric dipole because we expect that both left and right hemisphere are more or less doing exactly the same. Based on the topographies, probably we have the same activity on both sides, so quite similar. So it's not something that is in one hemisphere or in the other. So we apply these 2 symmetric dipoles. And here we can see that basically in all the medial temporal lobe, both left and right sides have a very good units of fit higher of 0.9 right here and here. So we don't have just a single point that explains very well all our topography, but actually we have a region, probably somehow also supporting our previous results, that we don't have one single area, but we have a structure, a network of different structures contributing to our component. So very briefly, the conclusions of this work is that we have applied blind source separation on MEG, and we have found one component that is quite robust because we found it in all our patients. And also this component is modulated by the task of memory recognition. So we compare old and new images, we have some modulation of the activity of this component. Moreover, the source of this component is located in the medial temporal lobe based on the dipole localization of its topography, as we have seen at the end. Moreover, the activity of this component is better correlated with those activities in structure related to the medial temporal lobe, the amygdala, renal cortex, the hippocampus. Then we have applied the partial correlation analysis, and we have confirmed that this component represents a network as both as the hippocampus, temporal pole, and the renal cortex has information that is unique on these areas, and they are part of the independent component that we have acquired on MEG. So we have confirmed finally that the hippocampus and also temporal pole and the renal cortex can be detected by MEG during cognition. But it still remains, how can we reconstruct their time series independently of the other co-activated neural sources? So how can we reconstruct the time source of the hippocampus independently of the temporal pole and the renal cortex? So this work has been done in the Institute of System Neuroscience in the University of Marseille in France. And thank you for watching this video, and if you have any doubt, please come to our poster and I will be explain and solve all your doubts. Thank you so much.